this will be recorded. The recording and the slides will be in Genetic Alliance's webinar archive. This is part of our registry bootcamp series. We are preparing a very robust bootcamp for anyone who's interested in a registry, and then particularly for individuals who are using our promise for engaging everyone responsibly. I'm Sharon Terry. I'm the president and CEO of Genetic Alliance. And again, we're happy to entertain your questions. If you would put them in the chat as Anne goes through her talk, that would be useful because we'll be able to have a little time at the end for conversation. So again, to remind everybody, you are muted. Happy to have comments and questions in the chat, and we'll take care of those as we go toward the end. So I'd like to introduce Anne Pariser. I've known Anne for actually quite some time when she was at FDA. And in the Office of Orphan Products, and now in her position as Director of the Office of Rare Disease Research. That is at the National Center for Advancing Translational Science, what we all call NCATS. And she's had a passion for rare diseases and particularly natural history studies for a long time. So I'm really delighted that we were able to get her to come and speak to us today. This is being recorded, as I said, just so that everybody knows that, for the archive, but the chat is not going to be available with the recording, so feel free to put any questions and comments there. And without further ado, Anne, take it away.
Well, thank you very much, Sharon, and welcome everybody, and thank you very much to the organizers for inviting me to Registry Bootcamp. So just to try to minimize any possible bandwidth problems, I'm actually going to turn off my video, and I can turn it back on again later during questions. So as Sharon mentioned, I'm going to be speaking during this presentation about the importance of natural history studies. So I'd like to just go through a brief introduction and brief rare diseases background, and then we'll talk more about natural history studies definitions and their uses. We'll discuss a few resources that are available to you and just a few words on data, and then I'll summarize a few of the key points. So as Sharon mentioned, I'm currently at the National Institutes of Health, and I think as many of you are familiar, this is the US's medical research agency. We are a public agency. We're part of the Department of Health and Human Services, and our mission is to try to turn discoveries into health. So what you may not know is that NIH is the largest public funder of biomedical research in the entire world. This year's budget was $42 billion, but actually got a little larger of that with the current public health emergency. And NIH is actually a collection of institutes and centers. There are 27 different institutes and centers, and many of them are clustered around a disease area, like the National Cancer Institute or the National Institute of Allergy and Infectious Diseases, which has been in the news quite a bit lately with Tony Fauci. But some of the institutes are clustered around, for example, a body part or body area, like heart, lung, blood, or an eye institute. So NCATS, or National Center for Advancing Translational Sciences, we are one of these 27 institutes and are actually the youngest institute. NCATS was established in 2012, and it's the only NIH center that is focused on translational science. So what do we mean by translational science? Well, translation is the process of turning observations that we could see in the lab or the clinic and trying to turn these interventions into interventions that actually improve people's health. And NCAT's mission is to try to move from the left side of the screen as efficiently as possible towards the right side of the screen. So to try to answer those critical research questions that are able to transform this research process into new treatments and cures that can be delivered to the patients faster. So our patient actually is the research process itself. And we don't focus on any one particular disease, but rather trying to improve the research environment that would benefit all diseases. So this makes us very unique in NIH. And I'm specifically from the Office of Rare Diseases Research, and our mission is the rare disease research process. So again, we don't focus on any one particular rare disease, but trying to improve the environment for all of them. So natural history studies are something that is very near and dear to our heart. So to just try to provide a little bit of background on rare diseases that I think may be very familiar to many of you, but the scope of the problem is really a large one. So again, as many of you are familiar, rare diseases are defined as a disease or condition that affects fewer than 200,000 people in the United States. But the reality is that most of them are far less prevalent than this. They affect only a few hundred or a few thousands, or sometimes less, single digits or double digits. So that rare disease can be defined differently in different parts of the world, but for the most part, most of these diseases would qualify as a rare disease really anywhere in the world. And there are about 7,000 or so different rare diseases. We don't know the exact number, and this number is increasing by 200, 250 newly discovered diseases each year. The diseases are not new. Our ability to recognize them is, and that's why this number keeps increasing. But rare diseases is also a misnomer. Rare diseases, while individually rare, collectively they're not rare at all. They affect about 25 to 50 million people in the US, about 350 million worldwide. And because of this, because of their rarity, because of how many diseases there are, and especially the low prevalence disorders, the majority of these are not well understood. And as a general rule of thumb is that the lower the prevalence, the fewer patients there are, the harder they are to recognize, the less well that we tend to understand them. And right now, only about 5% of our diseases have an approved therapy, but this is also a time of just tremendous opportunity. There have been many advances in science, particularly around genetics, and we now have the opportunity to really alter the course of rare diseases through things like gene therapies that are increasingly becoming a reality, or other highly effective therapies. And this gives us now a sense of urgency. We would really like to try to move as many of these diseases through the research process to get an effective therapy as we can. So this is a graphical representation of the diseases over time. So the left side of your screen starts in 1989, a little over 30 years ago. And at that time, we understood the molecular underpinnings or the molecular basis of only about 10 or so diseases. And I think you can appreciate over these past 30 years, largely due to understanding genetics and human biology, we now understand the molecular basis for about 6,500 of these diseases. And I think, as you can also appreciate, this is increasing by about 200 and 250 diseases a year. But again, there's only a small number here that have an approved therapy. And also, just to represent disease prevalence, and here's another graphic. This is actually from an Institute of Medicine report from 2010. And so if you remember from the previous slide, we recognized about half as many diseases as we do now, but the numbers don't really matter. It's really more the concept here. So towards the left of the screen again, well, the graph here is representing at the time the number of diseases that were known, and this large bar to the left, those are rare diseases, and the rest of these are common diseases. There are quite a quite a bit fewer diseases that are common, but there are a lot of people that have them. So then if we take these higher number of rare diseases and we look at it the same way and try to break it down in prevalence, we see the same thing, is most rare diseases are actually very low prevalence. They affect less than 3,500 people, or often a lot fewer, and there's just a lot more of them. And so this is really the challenge here for rare diseases. Sometimes these low prevalence disorders, sometimes called very rare, but most rare diseases are actually very rare diseases or ultra rare. So that brings us then to a little bit of background to try to frame why natural history studies are important. So now we'll move on to talking about natural history studies. So just start with a definition. This is a registry boot camp, and registry and natural history studies, sometimes these terms are used interchangeably. But when we refer to a registry, what we're really referring to in the broadest sense is an organized collection of data. And if we're talking about a healthcare registry of some kind, we're referring to a collection of information about individuals that are usually focused around a diagnosis or condition or disease or a group of diseases. So that's a very broad definition. A natural history study is actually a type of registry, and it is a pre-planned observational study that is intended to track the course of disease and to fully describe the disease, or as fully describe it as we can. So also representing this graphically, if the large purple circle is the world of registries, here are some examples of types of registries. And if you start at the right side of your screen, these can be some very straightforward, simple registries, such as a contact registry, where you're just collecting people's names and maybe their email address so that you have a way of getting in touch with people, disease information, or uniting your community, or things like listings or catalogs. For example, there is a registry of registries out there. Towards the other side, these are registries that generally need specialized knowledge or complexity or equipment. So at the bottom, there's this arrow going from the lower cost and complexity at the right to the higher cost and complexity on the left. But the important point here is that these are all useful and they can all help. And a natural history study is a specialized type of registry. And what it's mostly trying to do is collect this phenotype information in the middle. So that would be the clinical characteristics, as what is manifesting in patients, clinically, laboratory, and trying to describe that as fully as possible. But it can overlap with some of these things, and it depends. Some of them biobank, some of them include patient reports or a contact registry or imaging. So they can look different, but the core principle here is to try to fully describe that disease. And then just to briefly mention the types of natural histories, they generally have two elements to them. The first is it a prospective or retrospective study? So a prospectively is the data doesn't exist. We need to go collect it. So I can collect it now or moving forward, or both. A retrospective one is the data already exists. So this would be most commonly, it would be data that is collected for another purpose, for example, medical care. And I may go back and do a chart review, for example, and try to gather data on patients with one disease or a disease area to try to then get that organized collection of information to better understand it. The other element is that these natural history studies are generally surveys or cross-sectional studies, or they're longitudinal. So a survey would intend to collect data at one point in time, gather the data just once, such as a census or some of these spot surveys that you may have completed that aren't intended to be gathered multiple times, or longitudinal, gathering the data more than once. This could be a second look, perhaps at a point in time, or in some cases maybe planned to continue collecting data at multiple intervals. So natural history studies can be a mix of these, or they can change over time. These are observational studies. They're meant to describe, so you can change them as you gather information and you learn. So not infrequently, for example, you can start with a retrospective cross-sectional chart review, a one-time look at a medical record to try to get a start to gathering this information, and then decide, once you have this information, that you then want to alter your protocol and then collect more information or different information, or even a subset of that information over time. But regardless of this type, they all require prospective planning. They all require a protocol so that we're doing this organized collection. So then that brings us to why do we need natural history studies? What is it that we're trying to accomplish, and how do natural history studies fit in this? So I took here a quote from the International Rare Diseases Research Consortium, and as the name implies, this is an international organization that was intended to help various rare disease organizations collaborate to try to speed rare disease research. So this includes patient groups, government organizations, industry, some others. And one of part of the big vision statement is written here. It's to enable all people living with a rare disease to receive an accurate diagnosis, care, and available therapy within 1 year of coming to medical attention. So as I mentioned before, right now we don't have an available therapy for all of our diseases, but this is something that we want. We want an effective and safe therapy for all rare diseases. And so how is it that we're going to get there? Well, to get an effective, safe therapy, we have to have our candidate therapies tested in clinical trials so that we can show that they're safe and effective and then hopefully lead to an approval. So these candidate therapeutics would have to be evaluated in a good quality clinical trial, which in regulatory speak is called an adequate and well-controlled trial. But to design our adequate and well-controlled trial, we need foundational science to inform this development. We need to know something about the disease. We need to know about the drug that they're studying. How is it we think it's going to impact on people? And to do that, a natural history study, which describes the characteristics of the disease, is often one of the most important elements, but also it's a place to start. So then we would start up here, gathering this data, and then work our way down the chain as we identify information or also candidate compounds. So since we're trying to end up at the end of the day with an effective and safe therapy, I just want to briefly mention the standards for approval in the United States because this is what we are working towards. And this is why natural history studies are important. So the best access to a therapy is an approved product. And in the US, the standard for approval is what's called substantial evidence of clinical benefit from adequate and well-controlled trials, so that we can show that whatever this drug is, or device, or biologic, that it's safe and effective for its intended use. So what does that mean? Well, clinical benefit, that's generally interpreted to mean the effect of the therapy that is meaningful to patients. So it's the patients decide what is meaningful to them. And we have to know what that is. Safe and effective, it's generally interpreted to mean that the benefits of whatever you're giving is going to outweigh the risk, and all drugs, biologics, devices have some risks, some side effects. But it's also generally accepted or that we're talking about serious diseases, people will accept a higher level of risk. But can we manage those risks? We need to know what they are. So to put that in context, we have to have an understanding of the disease, what's important to patients, and also, what we can do to mitigate that. And then adequate and well-controlled. So what that generally means is that this is a study that is designed well enough based on accepted scientific principles, and that there is sufficient detail that we are able to analyze it and describe it, and it would then support the claims of effectiveness. So I will just briefly go through the elements of an adequate and well-controlled trial. And there are 7 elements here, and I'm going to go through them because this is a natural history study is basically going to try to inform many of these things. So every study should start with a prospectively stated hypothesis. You have to state what your goals and your objectives are so you know how to design your study. And so for example, I want to see if people taking drug X live longer than people who do not get the drug. And I would need a valid comparison to a control. If your drug works, how do you know? What are you comparing it to? So this could be a concurrent control or it could be an historical control. So for example, a placebo or another therapy, another active therapy could be a concurrent control versus data that has already been collected from the same population, which would be your historical control. We have to appropriately select the subjects, people with the disease, but people who are also reasonably likely to benefit from whatever you're doing. And we have to have methods of assigning people to a treatment or a control, such as randomization, measures to minimize bias, such as blinding techniques. And then importantly, we have to have well-defined and reliable methods for assessing the response. So what are your outcome measures. What are you measuring to tell if your drug works or if it doesn't? And then at the end of the day, we have to be able to adequately analyze this. So this is some of the things what a natural history study is going to put into context and inform us. So I'll come back to that in a minute. So also just to give you a very general overview of a clinical development process. So this is a traditional paradigm, and you may have seen lines like this, and with it basically trying to tell us that we start over here in our basic research labs and we gain our knowledge, and then we work through the translational process into pre-IND and then clinical search, and then at the end of the day we get our approved product. So you also may have heard that this is often described as taking 10 to 15 years, sometimes a little longer, sometimes a little shorter. But there's a lot of things that need to be done. And there's implied here is this linearly going from left to right. But the reality is things are a lot more complicated than this. So this is what's called the 4D map, which stands for Drug Discovery, Development, and Deployment. And this was actually a project that was initiated by the Genetic Alliance with the goal of trying to describe what is everything I would need to do to develop a drug. And it became this map, and it was published in 2 places, and the links are listed here. And right now it is up on NCATS's website with the link down at the bottom. And it's actually now a dynamic model, and you can click on various places in here and it will tell you more about what needs to be done in that area, as well as how it connects with some of the other parts. And you will notice that natural history studies actually appear twice. And they also appear in the middle, which is appropriate. So they appear down here towards this. This is leaning towards clinical development and the clinical side at the bottom. Like how do I design my trial? How do I find my patients? Or the one at the top. This is towards things like biomarkers, outcome measures, but also sitting in the middle of some of the translational science that needs to be developed. So to put this in another way is, rather than this linear model, drug development is really much more of a foundational model. It's like when we construct a house. You don't start putting the roof on. You start by digging a hole, building the foundation, pouring the concrete, the steel beams, because if you don't do that, the whole house is going to fall down. So it's kind of the same concept, as we have to build from the ground up so that by the time we get to clinical development, we have a really good foundation of knowledge, and hopefully try to minimize surprises. Surprises are bad in trials. So here are some of the things that goes on, and starting from the bottom. So always a planning. Planning is always very important. So the first thing is identify the disease or the disease area for study, or is there existing information that we can build on? So you could do one disease, you could do a related disease area. These all do fit within one study if it's laid out properly, or you could do even a subset of a disease depending on what you're studying. And then the intent again would be to try to describe the full spectrum of the disease. So very important to include a broad spectrum of patients by age, by geography. Many of these are genetic diseases, so they can vary by age. Generally speaking, the younger you are with a genetic disease, when it manifests, usually more rapidly progressive. That's not always true, but generally speaking. So you would want to represent the whole spectrum, from rapidly progressive to the more attenuated forms. And geography is also important because genetic diseases, again, often you see certain mutations or phenotypes of the disease that would cluster in a specific area. And then we would want to describe the things that are important to patients. So again, that goes back to the clinically meaningful benefit I was talking about before. So what are the clinical characteristics of the disease? What are the signs and symptoms? What are the lab values, imaging? How does the disease progress? Is it chronic and progressive, or is it episodic and people are relatively well between these episodes? And does it progress very quickly, weeks to months, or over decades? And there can be variations between. And then from this type of information, we can identify what it is that we need to measure, or can we measure. And there may be some aspects of the disease you can measure easily and some not as well. Do we need to develop assays? Do we need to develop tools? And then at the top here, how are we going to apply this information? We're providing the context of the disease, potentially comparators for the disease that will help inform many aspects. And to put this in the context of clinical development, as referring to before, so again, down at the bottom, the full spectrum of patients. Are we going to include all these patients in our trial? Or are we going to identify populations of patients who may be more suitable for this particular study? Are there some patients or information we could use here that would serve as a control group? Are we going to enrich our trial just with patients perhaps more likely to respond or who progress more slowly or rapidly? So these would all be decisions we'd make based on this context. And then out of this disease course, clinical manifestations, this is where things like outcome measures come from, biomarkers, clinical outcome assessments, but also the design of the study. Am I going to run it for a year? Is the disease progress enough I could measure something in a year? Would I need 2 years? Would I need 6 months? So hopefully we would have all of that information. And then again, at the end of the day, this interpretation, what we were referring to before with risk benefit. I have to put this in the context disease. It's a serious disease. Do I know it's a serious disease? Do I know that the majority of people are going to suffer serious consequences from that? And will it help me at the end of the day interpret this when I'm considering the side effects? And again, is who am I comparing this to? So then just to state this a little bit differently, so then when I've gathered this information, how then can I use it? Well, so the primary use, which I've tried to communicate to you, is the primary use is going to be to try to inform this clinical development across every stage. So starting from clinical trial readiness, these endpoints, outcome assessments. Can I design my trial? I know who I want to put in my trial. I know how long to run the trial. I know what to compare it to. And then again, I'm able to interpret the results at the end of the day. Nothing ever works perfectly. Is there enough benefit there that I can justify the exposure? But there are other uses too. So early on, natural history study can be used to unite the patient community. Perhaps this is one of those newly recognized diseases and we don't have a research program together yet. So this is a first way often to try to bring people together and to establish patient leadership. It's very important that patients control their data. It's also a way to collaborate with investigators and clinicians in industry. A well-organized registry and organized patient community is a real asset for a clinical development program, and this can sometimes be a deciding factor in deciding which programs to move forward with. It's also a way to begin a research program as we understand the disease better, perhaps identify targets. It can help put us on the road to drug discovery. And it can also provide a framework for diagnosis or patient identification. And I've had people tell me this many times. As they begin to describe the disease and they set up the registry or the study, they'll have people recognizing themselves and say, I have those symptoms too. So it can be a way to grow the community, but to help move people to diagnosis. And there are other uses too, and I'm sure you are aware of them. So I'd just like to say a word here about historical controls because, again, this has been described to me by people. They will describe it as, you know, this is a pot of gold at the end of the rainbow, or this is the holy grail of a natural history study, is that I could use this as that control group. So as I mentioned, this is not the most common use of natural history study data. The data can inform every layer of clinical development, and it's usually not the primary purpose of the study, although sometimes it is. And the purpose of any control really is what you're trying to do is measure what would have happened if I hadn't intervened. So there are some circumstances where an historical control can do that, but it's usually reserved for special circumstances, and this would be the minority of cases. So the disease has to have a high morbidity or mortality, and it has to be predictable. So we use a cancer example again. Advanced cancer that is not responding to traditional therapies, well, we know that has a poor prognosis, and we know generally what the outcome is. So that may be one situation. The effects of the drug need to be self-evident. They need to be very clear. They need to be objective. They need to be able to verify it. So again, mortality is something that is frequently used when an historical control is used. And it needs to be a fair comparison to your population. So natural history studies can go on for a long period of time, but medical care changes a lot. And so if your study data is 10 years old, it's possible that even in the absence of a disease-modifying therapy, clinical care may have advanced to the point where your 10-year-old data is not really fair to what's going on. So these are all things that need to be considered. But again, the important point being, it's not the most common use of the data, and natural history data is useful for many ways, not just a historical control if it cannot be used for that purpose. Okay, so now I'd like to switch gears a little bit and talk about some resources. So this little graphic here is, I'm going to tell you about some resources. There are many resources out there, and more than one road leads to Rome, but we're just going to share with you some of our experience at NCATS and ORDR. So our philosophy at NCATS is that natural history studies and registries, they're useful. They're useful in any number of directions. They can be useful for a small area or a much broader area, but they can all be useful. And we also believe that every rare disease should have a registry and or natural history study. And by A, we really do mean one. These are small populations, and if at all possible, it's better not to fragment an already small community over multiple studies. And the natural history study should be of good quality. And this goes back to what we're talking about with the planning aspects of it, but also the data, planning for data. And approaches that we're taking should be relevant and scalable to all rare diseases. Now, that's not to say that every natural history study is going to be done the same way, because it's not. It depends on the disease. But are the principles, the tools, the thought process, that can be reproducible and should be relevant to thinking this through and designing your study. And data is most useful when it can move, when we can share it, when it's interoperable, it's trustable, findable, analyzable. We'll come back to that in a little bit. So this is our general philosophy for how to proceed. So just to give you a little bit of history on this, so natural history studies are research. And research, more often than not, it doesn't work out the way you're going to think it can. But that doesn't mean that we can't learn from it. So back about 10 years ago, ORDR started what is called the Global Rare Diseases Patient Registry Data Repository, or the GRDR. And this was a proof of concept pilot project, and its aim was to develop a web-based resource that would design, house, aggregate different registries for rare diseases all in one place. So this was a very ambitious project. It also wanted to validate and implement common data elements. And also at the time, 10 years ago, natural history studies weren't as accepted as they are now, and to try to gauge the interests of the rare disease community. So they started with 12 patient advocacy groups who were going to build their own registries. And this ran initially from 2010 to 2013. And it succeeded, and it built 12 registries for these groups. But the problem is that everybody was off working on their own registry, and there was no collective ways of approaching these. And what they found at the end of the day was the data was not very easy to share, so it wasn't movable. So then a second pilot was launched for 3 years, where they took the existing data and then tried to map it to available standards and ontologies and languages, so that then the data could be more interoperable, and then we could learn the lessons from one disease to another. So after 7 years, take your 7,000 diseases minus 12, and we're left with 6,988. So it was just not scalable. It was going far too slow. We had too many diseases that we had to focus on, and this just was not proceeding fast enough for the way the science was advancing. And this post-processing of data, it was very complicated, it was very expensive, but also when you are applying standards to data that you already have, you can't change what was already collected. So it even with the mapping, it wasn't fitting together very well. And the common data element process is very labor intensive and it's very slow. So it was just decided that we just needed a better plan, and we needed to prospectively plan for better data collections prior to when we were collecting them. So that led us to, and this last one especially, we had to prospectively plan for better data. So that led us to a new approach, and we started a new program. It's called the Rare Diseases Registry Program, or RADAR, a bit of a tortured acronym. And this is a screenshot of the website, and here's a link down here. And if you came to the landing page, this is what it would look like. So this was launched in 2017. It's web-based, it's completely free, freely accessible, anybody can use it. And the primary audience we had in mind were patient groups who have decided to build a registry. So we wanted to be able to walk people through how to put a registry together, what you should think about, what are the tools that you need, decisions that need to be made. And we wanted to provide this community so that people could pick this up and get started. And within there, we're emphasizing things like data quality, standards, and sharing, and approaching this in what we're calling a tiered or a modular approach. So here's what we mean, starting from the bottom of this tiered approach. So in tier 1, these are data elements that everybody has. So everybody has demographic data. How old are you? Where do you live? How do we get in touch with you? So the beauty of this is that the common data elements already exist for these. There are standards, CDIS standards, for example, that FDA uses that you can readily pick up off the shelf and use, and that way everybody's doing it the same way. But these are also things that can fit into spreadsheets, Facebook pages, simple databases. So these are things that everybody can do. As you move your way up to tier 2, and tier 3, we're talking about a therapeutic area. So let's say this is collection of neuromuscular disorders, for example. Well, there's some diagnostic elements and characteristics that may be in common, and there may be some common data elements that exist for those, but there may be things that you can borrow from. But once you cross from 1 to 2, you're now in the realm of personally identifiable information, and that requires a big step up in things like data security, firewalls, computer expertise. And then tier 3, this is usually very rarefied air. It's very granular. It's about the characteristics of a disease. Usually the common data elements don't exist, and we still have those data security things. So as you move your way from the bottom to the top, the cost and complexity goes up and the ease of entry goes down. So often for above the red line, you're going to need a platform or some help. So this is just restating this again. Tier 1, a precedent exists. Use existing standards, search for what's available. Tier 2, getting that data privacy and security, seeking advice. Again, searching what's out there, or are there things that we can that already exist that we can adapt. And tier 3, this is often things that will have to be invented by who's doing them, that will be very specific for that disease. And we would urge a public process so that people are not developing data elements for the same disease, siloed efforts, and they aren't communicating. So how does RADAR address this? Well, the emphasis of RADAR is really on learning, trying to provide some tools and modules for those various tiers that I mentioned to help with planning and setting goals, and some tutorials on good data practices. And we've included things like downloadable templates. And importantly, though, is this, unlike the GRDR, this is not a repository. It is an information site. So this is a getting started page. This is what it looks like. These tiles here, if you click on them, they will take you to these various topic areas. So let's just use an example. So here's the set up your registry tab. If you came here to this page and create your registry plan, the first step, this is what the page would look like. And it walks through various topics here. And these blue words are links. So let's look at one link. This is a tool registry plan template. So you could download this. It's a PDF document. It looks like this, and it's actually 3 pages long, and it's trying to walk people through the questions they should ask and how to go about putting this together. So this is available on the website. I encourage you to come look at this, and if you're planning a registry, especially to work your way through the various modules. So then, as promised, I'm going to just say a few words here about data. And data tends to be a pretty dry topic. People often don't like talking about it, but it is critically important. So if you'll just hang in there with me for a few slides, and I promise I will give you some cute puppy pictures at the end. So I think everybody can agree we're living in an information explosion. Healthcare data is just increasing rapidly, and I just pulled a few of these graphics off the Office of the National Coordinator for Health Information Technology. These are public dashboards. There's many of them. They're kind of interesting to look at, but this one here at the top left, what this is just showing is the adoption of electronic health records, and you've seen it's just it's shooting up, but that's just using the health records. And if you consider that each patient has hundreds of patients, millions of pieces of data, and then multiply by what could be in one hospital system or medical system database, we just have vast amounts of data just in the EHR. But then we also have things like e-prescribing here at the bottom of the screen and how much that went up in just a 5-year period of time. But these are all, again, sources of electronic information we can look at. And this is something from a McKinsey report showing DNA testing adoption going up logarithmically, telehealth. So these are just vast amount of information. But while the amount of data is vastly increasing, our ability to use it isn't always going on in parallel. So just one example here, and it's not meant to pick on anybody, but just the electronic health records, this has been in the news, is you have all these different vendors, but the health EHR, they aren't interoperable. It's very difficult right now to share information from one EHR to another, and that's a problem. If you're seen at one hospital or your doctor's office and then a different hospital, it's not so easy to share this information, which, you know, unfortunately is what EHR was supposed to address. So just because the data is there doesn't mean we can always use it and we can pool it. So what is really needed is better infrastructure to support this, and there's just an incredible amount of work going on right now. But just one thing I would urge you is that about 10 years ago, a community of scholars, librarians, archivists, data scientists got together, and they put together what's called Force 11, and they came up with these FAIR data principles. And what this is to try to enhance the ability of us to be able to use this data, and particularly about the metadata, which is the data about the data. So is the data findable, accessible, interoperable, and reusable? And this is a topic onto itself, and I won't go into it any deeper, but I just did want to urge all of you, if you're thinking of starting a registry, you've started a registry, you have a registry, please spend some time on these FAIR data principles, because how we collect the data then enables our some to then use it later on, and it's just critically important to any data source. So as promised, after that, here's some cute puppies. So we'll just take a deep breath, and now I'll talk about a few additional resources. So a few years ago, last year actually, FDA published a Rare Diseases Natural History Guidance. This is a very good document, and it actually explains a lot of what I've just been talking about, and it's very readable. So I would urge you to take a look at that. Then towards the right side of the screen, this is the Agency for Healthcare Research and Quality, or AHRQ or ARC, and they actually have this is a reference guide, and this is a 2-volume weighty tome that goes into considerable detail about registries, and it is an excellent reference that really goes across almost any aspect of registries you want to know. It's not specifically for rare diseases, and it is available online. It's an ebook, it's downloadable, and it's free to anyone. So I would urge you to consult that also as a reference. And just a few more, as Sharon mentioned right at the beginning, but the Genetic Alliance has their peer program, which is a platform for constructing registries in a quality way. Here's a link. I think many of you already know about this. And a few other documents. FDA has published Rare Diseases, Common Issues and Drug Development Guidance, which goes into some of the things about natural history, but also clinical development process that is also very good. And the Office of Orphan Products has offered twice now natural history grants, and they have some information up on their site. And at NCATS, we also have what we call the toolkit for patient-focused drug development that kind of mirrors this FDA guidance. It's about how to guide patient groups through putting together their research programs. So it has a number of useful sources of information there. And then finally, I'll just mention the Rare Diseases Clinical Research Network, which is a research consortium run out of NCATS, ORDR again. And we include in this network about 250 different rare diseases. So before you start a registry, if you have a registry, I'd urge you to take a look at the list of diseases there, because a requirement of the program is they have to run natural history studies. So there are 250 diseases there that actually have ongoing natural history studies, and it may be possible for you to take part in that. So then I'd just like to summarize a few of the key points. So the key point number one is that foundational science is of particular importance to rare diseases. Rare diseases are poorly understood. We need this information for clinical trial planning, design, interpretation, but also for planning for research studies. So I just took here a quote from Janet Woodcock, who is the director of the Center for Drug Evaluation and Research at FDA. And she said that success meaning getting an approved product depends less on novel trial design than on the knowledge that underpins the program. So once you have that knowledge, it's a lot easier to decide on the trial design, but that knowledge is just critically important. And that's where natural history studies really come in. The use of natural history study data. So what we are advocating is one collection, many uses. Once you collect the data, it's recyclable. You can use it in a clinical trial. You can use it in setting up a program, an outcome measure, validation, or development. So it's about the disease. So you can use it over and over. And ideally, there would be this natural history study for one disease. But if there is more than one, or let's say you have a biobank and a natural history study and a contact registry, it's some way to seamlessly make them interoperable, then we will have much better use of the information. But in addition to therapeutics research, there are all these other uses that we talked about before. And then finally, data standards, interoperability, transparently are essential, and we've got to pre-plan for the good quality data. We have to keep the oranges with the oranges and the apples with the apples, and not get like an ORPL at the end of the day that you can't really use. And our puppy friends here are just trying to remind us that please do spend a little time with the fair data practices and understanding data practices, because at the end of the day, that'll help us get maximum use out of this very valuable information. So that was my final slide, and I just want to end by saying that, you know, in CATS, we collaborate with everyone. We're always very glad to talk with you. If you have questions, you can email me, you can call us, we can set up a conversation. So please don't hesitate to get in touch. And I'll stop there.
Great. Thanks very much, Anne. That was an excellent tour in a short time and some awesome puppy photos. So there's a couple questions in the chat. So a question, and I think these two are kind of related, so I'm going to put them together. So will you also discuss the potential for genotype-phenotype studies in orphan disease populations? It's an issue that's difficult to convey to NIH study sections when one is trying to get funding. And then the next question that I think goes with it is, due to the critical importance of these natural history studies, how come it's so difficult to find grant programs to support natural history studies?
Yeah, those are great questions, and I'll do my best to answer them. So I think the genotype phenotype relationship is something that is becoming studied much more. And partly it's because it's now easier to do them. It's just just about everybody is being genotyped now, particularly in rare disease studies, and as we understand the disease more, trying to correlate them. So I think that is ongoing work, and we've been seeing this get better even in the last couple of years. So hopefully that trend will continue. In terms of funding, yes, that's a very difficult question. It's rare diseases generally don't get enough attention and funding, and we are trying very hard to change that. But part of the problem with natural history studies is they're observational, so they aren't seen as innovative, so they don't always score well in study section, but also they're open-ended, and so it doesn't fit well in a lot of these grants programs. So unfortunately, I don't have an answer for you, but I think as we continue to demonstrate the importance of these, it gets easier to make a research case for it.
Yeah, and I think the other thing that I've noticed is as pharmaceutical companies have lost their blockbuster model, all of them have opened rare disease research and development arms, which at first we were all thrilled with, and now we see that they're trying to, disparaging remark here, do this so that they can replace the blockbuster model, perhaps with lesser numbers of drugs sold, but for higher prices. Be that as it may, more of them are interested in preparing populations. And so there is some funding sometimes for natural history studies through pharmaceutical companies. And those can be structured, I think, in good ways. Genetic Alliance has some templates for doing that around data sharing and around what gets back to the patient community at the end of the day and what can be used by more company than one and those sorts of things. Another question from your experience or expertise, can a natural history data collection effort be successfully separately outside of a patient registry organized by a patient organization?
Be successful separately outside of a patient registry organization? Yes, because you know, the registries can focus on very different things. So again, they can both have value. If these can be made interoperable, all the better, but I mean, they both can succeed on their own. They just can be more valuable. So for example, a natural history study done in an academic center by an academic investigator, they may have specialized equipment or specialized things that they're looking for that they may be able to study that a patient group couldn't, but the information that the patient group is collecting, such as what is important and meaningful to patients, that is critically important too. So I would urge you to try to collaborate if that is possible. But we also want to see patients have leadership and ownership of these data. It's your data, and you use this to decide how you want to put it to best use.
Okay, there's another question about IRB approval for a natural history study based on the survey of parents and their children's symptoms. This person's part of the Timothy Syndrome Alliance and hoping to write a natural history study, but because they're an advocacy group, they don't have an IRB connection. I'm going to toss that to you, Anne, but I'll just say Genetic Alliance established in 2003, so 17 years ago, an IRB exactly for this purpose. That's very inexpensive and very quick because we advocacy groups needed one. And then Anne, any other ideas you have about that?
Yeah, that was a great answer, Sharon. Is if you're collecting patient-specific information, you really do need an IRB. So there are some, such as Sharon's platform. There are some other academic institutions that can help with this, and there are some IRBs that will give you discounted rates because you're a rare disease patient group. So do look around, talk to people, talk to Sharon, but it is possible to get a relatively low-cost IRB, or if there's an investigator that is willing to take this on at their institution.
Yeah. And another question, how about population size in terms of natural history data? Are there any studies you're aware of that look at specific timelines and patterns of migration within the rare disease? Michelle, we might ask you to unmute and tell us, unless that's clear to you, Anne. I'm not sure what patterns of migration means.
Well, I can, or
Go ahead.
Michelle, go ahead.
Sure.
I mean, there's a whole area of study now on genetic epidemiology where they're showing, you know, how populations came out of certain geographic areas, and depending on what was going on in that geographic area, for example, high altitude or certain diseases, how these mutations sort of develop over time, which can be very good for the environment that you're in, but if you're taken out of your environment, then it does then become, you know, quote, a disease. I think this is one example. One obvious example is people coming from the African subcontinent, where there are certain infectious diseases, and then you come to the United States and those threats aren't there anymore, and you don't have the survival advantage, and you instead see certain diseases like sickle cell, but that's not the only one. There's a whole field of study there. Do I know of particular natural history studies studying this? I don't, partly because natural history studies are mostly focused on describing the disease, although I think that this is certainly a very interesting area. But Michelle, if that doesn't answer your question, please feel free to clarify.
Great. Anne, for a patient advocacy group looking to start a relationship with you and your team, what's the first step? Do they need to have a plan for a registry prior to reaching out, or be at a certain step in their planning?
No, absolutely not. Just send me an email and we'd be happy to set up a meeting with you to discuss it. I would encourage you to go to the website and the guidances that I showed, because that can really help you get thinking through the planning process, which can take a certain amount of time. But yeah, sure, just send me an email and we can set something up.
Great. Let's see, anything else? A question about discounted IRBs. Genetic Alliance IRB is the cheapest one I know. I set it up because I was tired of paying Western and university prices, and so it's very, very inexpensive. A person could pay it out of their own pocket. Okay, it looks like that's it, Anne. I'm going to thank you very much for all this information. Really robust. It goes beautifully into our registry boot camp. We will remind folks that everything that you said was recorded and will be available in the boot camp and in our webinar series. And then a reminder that Genetic Alliance also has a platform. We've built and run this since 1995, and we are really proud to say it's on brand new technology this year with a public benefit corporation, Luna DNA, giving us free technology for it, and have from soup to nuts solutions for all of this, including the IRB and all of the issues around getting the data ready. We very much believe what Anne has said, and I've learned from her over the years, so we're very happy to have our connection with NCATS. I served on the advisory council for some time, and also to continue to work as a community, as she said, over and over together for the right solutions. So thank you, Anne, and everyone have a good day.
Yeah, and thank you so much. Bye.
Bye-bye.